I'll start. Morena, no mai, haere mai, ki te hui Aotearoa, New Zealand Food Safety Science and Research Centre. Ko Vivian Hunt tēnei, ka nui te mihi nui, kia koutou katoa. My name is Vivian Hunt, and on behalf of the New Zealand Food Safety Science and Research Centre, I'd like to welcome you all here today to our, I think Kath said it's our 17th Spotlight on Food Safety Science seminar. And today our topic is Emerging Infectious Disease, a collaborative science-based approach to prevention and control, which is clearly a topic of great interest given the world we occupy at the moment in terms of this pandemic. And our speaker, Professor Nigel French, needs no introduction, but I am going to hand over to Kath McLeod, the Director of the New Zealand Food Safety Science and Research Centre, to introduce Nigel. Before I do that, I'd just like to remind you that if you do have questions, the chat has been disabled, but you can put them into the Q&A and we'll address those at the end of Nigel's seminar. So Kath, over to you.
Yeah, thanks, Vivian. Ngā mihi mahana kia koutou, a very warm welcome to you all. Look, normally Vivian facilitates this whole session, but today I requested the privilege of introducing our presenter, distinguished Professor Nigel French. Nigel's career in epidemiology, prevention and control of infectious diseases has spanned 30 years, and there have been many, many highlights and achievements made in that time. I'm not going to be able to cover them all here, so I'm just going to touch on a few that really resonate with me. Nigel's scientific contribution, first and foremost, has been very, very, very significant. So over 300 journal articles and 10 book chapters. He is held in the highest esteem by his colleagues here in New Zealand, and his reputation is recognised globally. As such, he's received 3 Massey Research Medals, the MPI Award for Significant Contribution to Food Safety. He's a valued member of the International Commission on Microbiological Safety for Food. Nigel currently sits on the Ministry of Health COVID-19 tag, for which we're all extremely grateful, and is a Fellow of Food Standards Australia, New Zealand. The New Zealand Food Safety Science Research Centre was initially established and led by Nigel for the first 3 years of its existence, and we have been extremely lucky to retain Nigel's expertise. So he currently serves as the Chief Scientist for the Centre. In particular, in the last 12 months, I have worked really closely with Nigel on a number of very topical issues, some of which he'll discuss with you today. And his experience, knowledge and dedication are second to none. There have been many, many weekends in which Nigel has worked to produce robust scientific data to support both the government and industry manage efforts around disease incursions, which unfortunately don't hold or respect public holidays, Saturdays or Sundays. So it's my great pleasure to now hand over to Nigel to provide us with his insights around collaborative science-based approaches to the prevention and control of emerging infectious diseases. So go for it, Nigel.
Well, thanks very much, Kath, and thanks, Vivian, as well. I'm just going to hopefully share my slides. So hopefully you can just see the big screen there. Is that okay? Great, thank you. Well, kia ora koutou. I'm going to just cover a few areas related to emerging infectious diseases. As Kath mentioned, it's something I've been working on for some time now. I'm going to give a bit of a historical perspective. I'm going to start off talking a bit about the COVID-19 pandemic, and then a bit of sort of general stuff around how and why infectious diseases, emerging infectious diseases emerge and persist, and some of the impacts that they have. A lot of focus is on viruses, but there are also some really important emerging bacterial pathogens as well, and these include some locally important ones too, such as Salmonella and Campylobacter. And then I'm going to circle back to talk a little bit about COVID at the end as well, to illustrate some of the new tools for prevention and control that we have in the toolbox. So first of all, everyone needs a definition. So the incidence, emerging infectious diseases are diseases where the incidence is increased. Usually it's defined within the last 20 years to threaten or threaten to increase in the near future. So these are ones that may well come around the track, so that we're worried about emerging. They're also defined as ones that are newly recognized in the population. So these might be novel pathogens that have not been seen before in human populations, or ones that are existing but are now increasing in their incidence and geographical range. So that's what we define as EIDs. So COVID-19, I have to say, you know, having worked on emerging infectious diseases for some time, I actually feel quite moved by what's happened over the last 18 months and quite disturbed in many ways by by how things have developed. Nearly 250 million recorded cases globally, and we're getting on for nearly 5 million deaths. I'm just going on the slide on the right-hand side. There's actually some wealthy countries, and just showing the impact that it's had. Biggest reduction in life expectancy since the Second World War. So these are the blue bars showing that reduction associated with COVID, comparable with the 1918 and 1945 reductions due to the First and Second World War. But it's also had its biggest impact, I would say, in lower and middle income countries as well. We can see huge inequities in terms of vaccine distribution. The graph on the left-hand side is from a Nature publication showing how the wealthiest countries have really had the highest proportions of their populations vaccinated, whereas the less wealthy are very, very few. We're seeing a big impact on the Sustainable Development Goals. That's one example there in terms of extreme poverty. The first increase in extreme poverty for several years, in decades. And then we're seeing the re-emergence of some other diseases as a result of COVID really dominating the health scene. So a reduction in testing resulting in estimated around about 500,000 new lives to be lost as a result of a very old disease, tuberculosis. And this is the global picture now. You can see these big waves of infection. This was only updated a few months ago, actually, but we're still seeing around about 500,000 cases a day. And importantly, in terms of deaths, and these are just recorded deaths, of course, between 5,000 and 15,000 deaths per day globally. So this is just of huge significance to the human population. And this is obviously the situation we're in New Zealand now. The first wave was significant, but was eliminated successfully. There were a number of subsequent smaller introductions that led to community transmission. And of course, now we're seeing the largest daily increase, 94 cases yesterday. And I'm really concerned about the impact that this is going to have, particularly on some of the most vulnerable populations in New Zealand as well. And if anybody's interested, the paper there by Sarah Jeffries describes the impact of the measures that we took during the first wave to eliminate that one successfully. Unfortunately, not been successful with the Delta wave. I'm just going to illustrate, this is the first wave, and then I'm just going to, and then right at the end of this little video, you'll see the emergence of the second smaller wave. This is over a year ago now. This was in August 2020. I'm going to use this just to illustrate how rapidly COVID can spread over the whole of New Zealand. But also on the left-hand side is a phylogenetic tree, and I'm use phylogenetic trees on a daily basis in the work that I do on a number of different pathogens. It's using genome sequencing, and you'd think about it just like your family tree or your genealogy. Isolates that are close, or cases that are close together in the tree, are ones that are more closely related to each other. And what you'll see when I show this is how whole lineages of COVID arise and then decline as we eliminated it through that first wave, and then you'll see another lineage emerging as we have the infection in Auckland in August 2020. So we can see this is the first wave. Now you can see multiple lineages, different strains of COVID that came in across the border, and then over a period of time were gradually eliminated, and then we successfully achieved elimination. We then see the odd case associated with MIQ, but not crossing over into the community. And then the big August outbreak that occurs, a single lineage that happened in August 2020, and that again was eliminated. So COVID-19 is really the latest and biggest impact that we've had from emerging infectious disease, and it did cause a pandemic. Now, not all emerging infectious diseases cause a pandemic, but when they do, they're clearly quite devastating. Now, this just shows the variety of different emerging pathogens that have happened just over the last few years. This is a slide actually from 2017, but it shows there's many viruses, there's many bacteria that emerge all around the world, some of them causing global pandemics like HIV, for example, and others which are much more localized, but they very frequently occur. I'm going to talk just a little bit about zoonoses, because zoonoses are pathogens that are transmitted between animals and people, so they can go one way or the other, usually from animals to people. And most of the emerging infectious diseases that we see are zoonoses. They come from animal reservoirs or maintenance hosts. Sometimes they're bridging hosts that then transfer them to human populations, and then they can eventually end up cycling in human populations. And over 60% of all human infectious diseases are these zoonotic diseases, so they're really important. COVID being one of them, of course, originally. Most infectious agents affect multiple hosts. These are animal infectious agents, are multiple hosts, and particularly the emerging ones. And a subset of these are the ones that go from animals to people. Some of them just go from animals to animals. And actually, single host agents are relatively rare, ones that only affect single animal species. And a subset of those are the human-to-human ones, so think of something like measles. And it's believed that the human-to-human transmission only really evolved once human populations had reached a size where there was sufficient fuel for those pathogens to be maintained in human reservoir populations. So as I mentioned, putting it in perspective, over half human infectious disease agents are zoonoses, and over 3/4 of them, of the emerging infections are. And also 80% of the weapons that are used for bioterrorism are also zoonotic pathogens as well. And they've had a huge impact. So we've seen global pandemics, we've seen associated with the H1N1 1918, we saw the swine flu emergence, we've seen HIV, and now COVID. And even pre-COVID, there were estimated to be nearly 2.5 billion infections, particularly in developing countries, that were caused by zoonotic diseases, causing over 2 million deaths. So they are hugely important. And this is just one way of understanding how they emerge. So we can think of them going through 5 different stages. So ones that are still only in animals. So this might be something like canine parvovirus, something that you vaccinate your dog with, hasn't crossed over into people yet. Then we have ones that cross over, but they still rely on their animal reservoir. So rabies is a very good example. It's a very old disease, not one that's emerged recently, but still causes significant fatalities, particularly in certain parts of the world. But you need a bite from an animal in order to cause infection. Then you have diseases like Ebola, where you get some cycles in human populations, and some of them can persist for quite some time, as we've seen. Then diseases like dengue, which are more sustained and long outbreaks, and HIV, where it's crossed over, and now we only see human-to-human transmission. So that's a stage 5 exclusive human agent. And arguably, SARS-CoV-2, the cause of COVID-19, is now a stage 5 zoonotic pathogen. So why do they emerge? And there's a number of things. I will talk about human risk factors, about animal risk factors, or livestock risk factors, and then wildlife. And there are many, many regions, and these are just some of them. So the huge increase in human population densities, tendency to converge in big, dense populations in cities, and population growth. We also see a lot of travel. This is a simulation of flights in a 24-hour period, obviously pre-COVID, but increased global travel moves pathogens very effectively around the world. Still persisting chronic poverty is also a major driver for the emergence of infectious diseases, and things like changes in dietary habits, so the consumption of things like raw food. So domestic animal factors, so expanding globalization of the food production system, poor biosecurity, poor animal health, and increasing antimicrobial use is driving the emergence, particularly of antimicrobial resistant pathogens. And then we have wildlife risk factors. We have a number of emerging zoonoses, including COVID, which originated in wildlife populations. Human encroachment into those wildlife areas is an extremely important driver for the spillover of these into human populations, and habitat destruction. Climate change, a really important driver. We've seen in the recent thing the use of wet markets, particularly wet markets selling wildlife. And it's actually been an estimate that the crossover of pathogens, even SARS-like coronaviruses, occur very, very frequently. It just happened to be that SARS-CoV-2 was very effective at causing a globalized pandemic. An estimated huge numbers per year do actually cross over. They just don't cause global pandemics. And we historically have been very poor at predicting when and where pathogens that emerge are likely to come from. This was a paper in 2008 in Nature, which tried to estimate where pathogens were likely to emerge from. And you can see the hotspots there were around Asia in particular, and also in Europe as well. But in fact, that was the year before swine flu emerged, and that was the one that emerged in Mexico. So it wasn't really predicted well where it was likely to come from, but that did cause a global pandemic, started in 2010. Pandemic preparedness, and I think this is something that is going to be a huge topic around, particularly after COVID. How best do we prepare? And I think this is a great quote: pandemic preparedness, telling governments what they don't want to know to spend money they don't have on something they don't think will happen. Well, I think now we know things like this do happen, so hopefully our preparedness in the future will be much more effective. And we should really try and learn from lessons of the past. And this really, if you, not many of us, I don't think probably any of us were around in 1918, but it's thought that the Spanish flu arrived just at the end of the Second World War, and I'm not sure this is believed to be true now, but I know there were some infected people on a boat that was bringing the Prime Minister Massey, back, and it's believed that some infections were set up by some of the crew members of that boat actually coming ashore and then spreading it into the New Zealand population, and it was devastating for New Zealand. And you'll see the similar things, overwhelming of the healthcare system. So we've got commercial vehicles being used as ambulances, a big impact on the most vulnerable populations, 8 times mortality in Maori, particularly during the devastating second wave of the pandemic. And also the use of controversial novel methods. I think of the use of some of the things that were advocated by President Trump, for example, that turned out not to be very effective. Well, in the 1918 pandemic, there was set up these zinc sulfate inhalation chambers, which rather than actually helping to prevent flu, was probably responsible for spreading the influenza virus amongst the population. And we also had controversy in 1918 around the implementation of public control measures. So obviously there was a lot of antipathy towards lockdown processes, but also masks. You can see this is an advertisement from the 1918 pandemic about against the use of masks. So that's an historical perspective, and obviously we should really try and learn as much as possible from these historicals. So history does have a tendency to repeat itself. I'm just going to talk a little bit about these global risk reports. So this is just the ones from the last 3 years. So this is produced by the World Economic Forum, and they're quite interesting to look at because what they do is produce a graph which shows what the biggest global risks are to population associated with economic, environmental, geopolitical, societal, and technological issues. And in 2019, climate change was right in the top right-hand corner, so it had high likelihood and high impact. Just by contrast, you can see in the top left-hand corner, weapons of mass destruction, obviously high impact, but very low likelihood. Spread of infectious disease was relatively low likelihood, but high impact at that time. Now, this was looking, this is a 2020 report, but obviously it was looking back over the previous year before COVID-19. And we saw infectious diseases still pretty much relatively low likelihood, but a high impact, and climate inaction still in that top right-hand corner. Now, not surprisingly, the latest one that we have shows that the spread of infectious diseases is now moved right into the top right-hand corner in terms of both likelihood and impact, alongside climate inaction. And these two are obviously related to each other as well, so we need to take this very seriously. So not just viruses, so we do also have emerging infectious diseases, which are really important bacterial infections that also arise as a result of foodborne spread. The big example that we have is the 2011 outbreak of an enterohemorrhagic E. coli, which was particularly focused in northern Germany. And it caused a really large number of cases, nearly 3,000 cases of bloody diarrhea, nearly 900 cases of a serious complication, this kidney failure, hemolytic uremic syndrome, and over 50 people died as a result of the emergence of this pathogen, which really hadn't been seen before, and had a number of epidemiological features which were quite of interest at the time. It didn't really spill over into a global situation, although a number of other countries were affected, particularly travellers moving outside of Germany. It took a little while to get to the bottom of it, but it turned out it was actually bean sprouts, contaminated bean sprouts, and probably the seed from those bean sprouts were contaminated with this particular E. coli that then spread and caused in the incubators of the bean sprouts, then got into the food, and the food was consumed and caused this issue. It took a while to find this out, but the case control studies and other things implicated other sources of food at the start, but eventually bean sprouts and the seeds themselves were implicated. Now, I just put this slide up. This is a timeline just to illustrate how we're not always on top of things. So there was a lot of congratulations about getting rid of this particular outbreak, but actually in reality at point A was the point of highest exposure. So that's when most people were consuming the contaminated bean sprouts as a result of the fenugreek seeds that were contaminated with the E. coli. B is the point of the epidemic itself. So it was obviously an incubation period before people started to get sick. But C was actually the point at which we realized, or which it was realized, that it was actually bean sprouts that were caused. So well after the pandemic, or the epidemic in this case, was over. So we're not always that quick to get on top of things. And in retrospect, we should have known, really, because more women in every age group, or females in every age group affected, a relatively long incubation, a lot of characteristics which were what were well known to be called sprout breaks in other parts of the world, but this wasn't really recognized at the time, so this may have helped to get on top of things. So what about, what about the emergence of our well-known other E. coli from a food safety point of view, E. coli 0157? And this is where there has been some work. This has been around a lot longer, and many different strains of E. coli 0157 have spread around the world, including into New Zealand. And this is just a piece of work that shows the importance of international collaboration, in this case, the use of genome sequencing on a global scale to try and estimate how and when E. coli 0157 emerged and traveled around the world. This is a picture from that particular paper, and it shows how it spread probably from Central Europe at the time to North America, and also to South America and to Australia and to New Zealand. And we can see estimated around about at least three or so introductions into New Zealand in the latter part of the 20th century. But we can also model this by using the genome sequencing and reconstruct the genealogy of these strains back through time to estimate when the ancestors were. And we can see that the first cases going from 1920s and into New Zealand 1960s, and still movements right the way through to the end of the 20th century and into the 21st century. We've also seen other bacterial pathogens that have emerged to cause devastating impacts, particularly in low and middle income countries. And this is an example of Salmonella, a non-typhoidal Salmonella, which is behaving like a Salmonella typhi. It's a strain called sequence type 313. It spread across sub-Saharan Africa and was associated with a really high mortality. And today, I don't think we've found any animal reservoir. It seems to be primarily crossed over and causing that kind of stage 5 human-to-human transmission, and is believed to be partially host-adapted now. I just mentioned this because this is a piece of work that was done as a really good example of collaboration. And this is collaboration between multiple organizations in New Zealand and internationally, with our Tanzanian, Kenyan collaborators, and also in Scotland as well. This was a project led by the Scottish team. And this was a project called the Hazel Project, looking at Salmonella in the food production chain. And it was a truly transdisciplinary approach. It involved everything from genome sequencing to social science work, looking at various different aspects of how these pathogens are transmitted. And it was very much a One Health approach. But just to point out here that we did detect Salmonella, that strain 313, but again, only in human cases from Kenya in this case. But many of the other strains of Salmonella, this one here, ST11, which I'll mention later, was found also in poultry and other sources as well, along with the other major strains. But 313 was found exclusively in the human cases. And it's associated with bloodstream infections and very severe outcomes. And these are two papers from that study. And again, I'm just putting these up to illustrate. This is a multi-institutional European, African, New Zealand, Australian collaboration, which we published a number of papers from this now. And I just shout out to Kate Thomas. You'll see the second author on the first, and also in the second paper here. Kate now works for MPI, and Kate was instrumental in setting up the laboratory in Tanzania and did a superb job with both the work on Salmonella and on Campylobacter in very difficult circumstances. So Salmonella, what about Salmonella in New Zealand? And we have seen our fair share of emerging and persisting strains, new strains of Salmonella that have arrived in New Zealand. One of the greatest examples, which has been well described and published, is the emergence of Salmonella typhimurium DT160. So it's a particular strain of Salmonella. And this is believed to be involved both wildlife transmission, foodborne transmission, and human to human, probably involved in the spread of this one. First human case was described in 1998, but it became the most prevalent serotype in New Zealand for over a decade after that. Caused a large outbreak in Christchurch in both people and also in domestic passerine birds, particularly sparrows and silverites. There was a big die-off of these particular wild birds around Christchurch, which spread over to the rest of the country subsequently as well. And then after 2000, it progressed from, it's focused around Christchurch right the way across the whole of New Zealand. So it was a very successful emerging pathogen that spread right the way from the south to the north. I'm just going to show a map of this. So this is a map of the human cases. I'm just going to illustrate this with two different strains. So the first one is a strain of Salmonella typhimurium called 156. And this is one that's predominantly in North Island in food and cattle, and it's been around for a lot longer. You can see that there are already quite a few cases in North Island and South Island. The blue one is really the early stages in 2000 of the first few cases in Christchurch. There's one in Invercargill as well. But the next slide just shows how rapidly it spread. And look at the timeline along the bottom as well. So we're moving just within 6 months, you're starting to see cases spread into North Island. And then over a period of very rapid spread, just within that first year to 18 months. And by the peak of cases in 2021, we're already beginning to see cases right at the tip of Northland all the way down to the bottom of South Island as well. So this is an example, whereas the other strain stayed relatively stable in North Island. So this strain's been looked at in quite a lot of detail. There's a publication in particular about it. The first case in 1998 was right almost at the fulcrum of the phylogenetic tree, so really indicative of this being very close to the ancestral strain that arrived in New Zealand around about that time, and then subsequently it's been evolving since. And we could show through genome sequencing and modeling that the population of this particular strain of Salmonella increased hugely. This is a log scale on the left-hand plot over those first 2 years, probably as a result of transmission with wildlife and also domestic animal populations as well, and crossover into people to cause this quite large epidemic sustained over a number of years with interhost transmission throughout. We can date the common ancestor of this to be around about the time of the first human case. So it really was a relatively recent introduction at the time. How do sparrows infect people? Well, we know from a case control study, handling of dead birds, but we also know things like sparrows also get into supermarkets, outdoor cafes, very good example in the picture there, contaminating potentially roof water, children's playgrounds. So there's a number of different ways in which we can get infected through wildlife, but it's also crossed over into the food chain as well. I would say subsequently it's also been replaced by another strain, and if anybody's interested in that one, DT56, is a paper just appeared this year describing the genomics of that particular new strain that took over once DT160 disappeared virtually. That's not the only strain of Salmonella which has emerged and caused problems in New Zealand. More recently, we've seen the emergence of cattle-associated strains. I haven't got time to talk about the epidemiology of these in detail, but they're really fascinating. They're Bovis morbificans and Salmonella give, in particular, are two strains of Salmonella which have emerged in New Zealand relatively recently, associated with cattle, also associated with an increase in human cases. So this is Salmonella Bovis morbificans in people as well. There's a lot of work to do on the epidemiology and control of this particular pathogen. It'd be interesting to see how this has tracked over the last year or so as well. And then one of our most important recent introductions is a new strain of Salmonella that emerged in 2019 in New Zealand. And this is a strain of Salmonella enteritidis. You may have spotted in the slide I showed earlier on of the work done in Tanzania that Salmonella ST11 enteritidis was also quite prevalent in that part of the country. It's a very common worldwide strain of Salmonella enteritidis. Through epidemiology, source tracking, and genome sequencing, ESR detected this strain. First of all, emerged around about 2019. Other sequence type ST11s had caused disease in people, but they were quite differently related. On the right-hand side, you can see quite closely related in blue is the emerging outbreak strain against a background of introduction of also ST11s, but not associated with this particular strain. It caused a big outbreak in an Auckland restaurant in 2019, 38 cases of which 17 were confirmed. And then it was detected in raw poultry meat in the National Microbiological Database, and subsequently detected in broiler feeder flocks, and also resulted in cross-contamination to layer flocks and down through the layer flocks. And to, again, result in some human infections as well. It was also detected in other species, we can see there, which is probably not surprising. Now, the control program was implemented by MPI with the poultry industry, and it looks to have been very successful. The Blitz survey covered 80% farms that result in the supply of 80% of table eggs, and the rearer and layer farms resulted in a test cull, sanitize, retest program, which seems to have stopped the tap of infection going through into people. We've had 48 cases in total in 2021, none very recently, and that's 115 in total since the start of this emergence. And we have used genome sequencing to get a handle on a number of things. So the date at which it likely came into New Zealand, to see where it likely came from globally, and also how it's been transmitted through, in this case, the food chain, the poultry supply. Now, this is the genome sequencing done by ESR, and just some analysis of those strains that we see. This is a mixture of isolates from poultry, some of those other animal strains, and also from human cases as well. You can see the Auckland outbreak in the bottom left-hand corner there, and you can see the distribution of cases over time. If we look in, zoom in a bit, at that top right-hand corner there, we can see that a lot of human cases in there. So the orange ones are human cases, but we also see cases in poultry, in the hatchery, in particular the light green ones you can see dotted around this part of the tree. A few mutations, these numbers here represent mutations in the genome, but generally the mixing of human with both broiler, layer and hatchery isolates suggests the transmission through the hatchery to layers and broilers and then on to people. This is a restaurant outbreak you can see in 2019, but importantly, it's believed that, well, if you do the modelling back to the earliest date of common ancestor, it's relatively recently emerged, so probably only arrived in New Zealand just before that outbreak that was in the restaurant. The recent transmission, there still was evidence of transmission in June, so we can see the cases here. This particular strain, substrain or sublineage with 5 mutations is found both in poultry environment and in human cases as well. So very good evidence, even in June, of some transmission between poultry and people. What about the global picture? So we took all of the isolates that we could download from internationally, which had a date and a geographical location, had a close look at these, and we could see the New Zealand strain. This is like a 2-dimensional map. So again, isolates that are close, or cases that are closer to each other, are ones that are more closely related to each other. And right in the middle there is the New Zealand outbreak strain called CO1. And you can see the strains from all over the world, some clustering in big groups in other places. Now we zoom into that one and we can see the New Zealand outbreak strain really tightly bundled in the middle there. And the other closest cases, interestingly, are from Europe and from, in particular, from Turkey, and also from the United Kingdom. And this is another interesting one. The nearest to the outbreak strain found in New Zealand is from a traveller who also had an association with Turkey as well. We don't know exactly, it's still quite different, but it'd be really interesting to find out where this particular strain came from. Campylobacter, a really good example of another emerging infectious disease that caused problems in New Zealand. Up to 2006, we saw this rise in cases, then the introduction in the poultry industry of interventions, which resulted in a big decline in human cases. Now, although there were many, many strains that were involved in the epidemic of Campylobacter in New Zealand up to 2006 and beyond as well, there was one strain that dominated, and this was quite an unusual one. It had only really been found once in Europe prior to it being detected in New Zealand. So this was really known as the New Zealand strain. It was called 474, sequence type 474, and it was very prevalent in those early cases. And revealing quite rapid evolution. We were interested in where it came from, why did it cause emerge, and what subsequently resulted from that. And we look, using genome sequencing again, you can actually look in quite a bit of detail. We find that actually it isn't one single strain, it's actually several different groups of strains that group together, and these are called clades. So clade 1, 2, 3, and 4. Clades 1 and 2 have been present all the way throughout and probably still around now. Clade 3 caused the first big wave of that Campylobacter outbreak between 2005 to 2008. And it was predicted to have emerged around about 2004, but hasn't been seen subsequently. Clade 4 emerged after the interventions in the poultry industry, but again, that one seems to have died out and has not been isolated after 2014. And interestingly, we can then look to see of the isolates that were in animals, so these are ones from animals or from the environment of this particular strain, that the epidemic clade was really strongly associated with poultry, and also the post-intervention clade, strongly associated with poultry. But what we see is a background of continual dripping of cases through, which are very strongly ruminant associated. So this suggests that maybe the strain that occurred in poultry might have crossed over from the ruminant population into poultry, expanded and clonally expanded quite rapidly into the food chain, into the poultry supply, and then causing human disease. And then after the intervention, subsequently disappeared, and we're now back to the strain largely associated with ruminants. So it's a very good example of a strain that most likely did cross over, cross species, and then caused an epidemic or an outbreak. But subsequent control measures have now virtually eliminated it, and we're now back down to the background strain. So an example of host switching and emergence with consequences for public health. So after 474, we then saw the emergence of yet another strain of Campylobacter, which seemed to be just really a New Zealand associated strain. And this arrived in 2014. And the interest in this one was because it was resistant, unusually resistant to 2 different classes of antibiotics. So the emergence of the new strain probably came into New Zealand around 2014. We don't know where it came from. Found quite rapidly in the North Island poultry supply, in the parent flocks, but also in poultry companies, actually all the poultry companies eventually. Big increase in antimicrobial resistance, not surprisingly, in chicken isolates, but also outbreaks in human cases, and a big increase in the cause of this as a cause of human cases in New Zealand. Again, we've used genome sequencing to show that it really was quite distinct to anything else that we'd seen before. 474, the strain was quite different. This one was very different to anything else we'd seen. So it's most likely had come into New Zealand and then spread through the poultry supply and then into people. We were interested in a whole number of things, such as how it emerged, what's the genetic basis for resistance, and all of those factors. So again, we did genome sequencing. We could show that it was evolving quite differently in different poultry suppliers in different parts of New Zealand as well. And from that, we were able to get an understanding of the importance of both local as well as within poultry supply spread as well. So public health significance of this, it was resistant to 2 classes, but they're not antimicrobials that are used even for invasive, more serious Campylobacter. Antibiotics aren't really recommended for use in Campylobacteriosis cases anyway. But we do know from the case control study that some of the one of the drugs that it was resistant to was actually being used or continuing to be used for Campylobacteriosis cases. It didn't cause an increase in notifications. We can see on the graph on the bottom right-hand side there. But I think, and it's in a case control study, around about 7% of cases, but we know it's still out there. And I think it does really highlight the importance of good biosecurity and reducing the risk of transmission between farms within poultry supplies and the risk of onward spread. So I'm just going to finish up with a few slides just about circling back a little bit to COVID as well, because I've talked about emerging infectious diseases and how they can have an impact locally within New Zealand and some of the bacterial EIDs that we've seen over the last few years and how they've been controlled. But just to point out that the obvious really, I mean, we talk about the team of 5 million, but emerging infectious disease and preventing and controlling pandemics, research scientists have played a role, but really, we really have to acknowledge that the vast majority of prevention and control is down to other individuals. So global health organizations, huge values in the World Health Organization, the healthcare workers, the frontline workers, the vaccinators, the samplers, politicians who are making very difficult decisions as we speak, community leaders advocating strongly for testing and vaccination, people working at the borders, other essential workers, supermarket workers, and other people that are in day-to-day contact with the population who can't isolate in the same way during lockdown. The investigators, contact tracers, people involved in surveillance, for example, at ESR and in the regions as well. Diagnostic laboratories, including the application of genomics, has been a huge effort. Modelers, epidemiologists, public health, all of these people, and obviously with vaccine and therapeutic development, a massive effort to preventing and controlling the pandemic. There's many others involved in this as well. R&D of new tools, and the obvious ones are vaccine, unprecedented, rapid development, approval, trialing, and dissemination of vaccines. It's not been perfect by any stretch of the imagination, and we're still seeing the struggle in New Zealand of getting the most vulnerable populations vaccinated. But we've seen a huge, simply wouldn't have been possible. We wouldn't have vaccines available just even a few years ago if we didn't have the technology that we have now. Diagnostics, modeling, and genomics, and again, a shout to the ESR team for the implementation of genomics for real-time sequencing that's so important for genomic surveillance. They're really developing that as part of their core work that they do for New Zealand. And that's also seen the advent of wastewater testing. And also, I'm just going to touch on this last thing here, which is the use of what we call phylogenetic modeling. Biodynamic modeling is just something that I've been involved with a team of people from ESR, University of Otago, University of Auckland, since the start of the COVID pandemic, really. This is a paper from Gemma Geoghegan, who works for ESR and for the University of Otago, just describing some of the genomic work that was done during that first wave. A high proportion of those cases were sequenced. And the study showed that using the phylogenetic approach, this was linked to, this is just looking at one of the clusters that's described in that paper, and it's the Bluff Wedding cluster, around about 100 cases. It really showed that the membership of this cluster, that all of those the cases that were really identified by the use of genomics were then clearly ring-fenced and identified as part of this cluster. It showed it to be a massive super spreader event. It also showed the impact of lockdown, the level 4 lockdown had on the R number, which everyone knows about these days. It went from around about 7 after lockdown to around about 0.2, so big effort, big approach. The alert level and lockdown had a massive effect on that R number. This is the Delta outbreak in Auckland. This is just showing one of the most recent phylogenetic trees. And what we can again see is this really widespread dissemination around, this is just looking at Auckland in focus there, but on the left-hand side, the phylogenetic tree, you can see the different epidemiological clusters, the different genomic clusters that we can see. And we see some, particularly the top one in purple there, is the one originally associated with the church super spreading event, which virtually disappeared, but then subsequently, unfortunately, we've seen evidence that were hidden chains of transmission from that particular outbreak that have then recently emerged again. And now the situation is we've got a different strain that's really prevalent, but also lots of mystery cases, these ones in grey here, that are still prevalent and causing problems. And being able to do this work now the case numbers is really increasing, is becoming increasingly difficult. One of the things we did do early on was try and use some of the modeling approaches combined with genome sequencing to get a handle on these mystery cases, to see if we could help inform the contact tracers and the case management teams to identify the likely sources of infection. And this is just an example from one of the reports that we were producing almost on a daily basis up until fairly recently. We could then look at individual cases and say, well, based on the genomic information plus the clusters that we know, these may be household clusters, they may be workplace clusters or whatever, and the onset dates, the symptom onset dates, we think that the most likely source of this particular case is this group here, and most likely these cases. And we combined this with modeling, which enabled us to also estimate the probability that it came from a particular cluster, and also whether there was an undetected chain of transmission. So this was something that was being done until fairly recently. So just to finish up with just my thoughts around collaboration. I mentioned this in the original title to this talk as well. And I can't stress this enough. My experience of collaboration, working collaboratively across institutions and in multi, inter and transdisciplinary approaches is really key to success in the implementation of research to inform decision making. And I really enjoyed working with a whole number of researchers across New Zealand, from the research institutes, particularly the ESR team for the COVID work, and other work on foodborne pathogens as well. Multiple universities, government agencies, Ministry of Health, MPI, industry partners, and also our international partners, which play such an important role in helping to advise New Zealand on different control measures. So collaboration really is key. So just to conclude, it's obvious from what's happening now that EIDs still represent a major and probably increasing challenge to human beings globally, particularly in the most vulnerable populations worldwide, from local outbreaks to epidemics and pandemics, so differing on spatial and temporal scales. The new emerging infectious diseases are coming on continuously, and the need for ongoing surveillance, so this is why the work that ESI is doing around genomic surveillance is particularly important for the early detection of these cases. We know that viruses and bacteria are particularly important, and it could be any pathway, so respiratory and enteric pathogens. And this really is the urgency to develop real tools that we can use to prevent and control in the future. It all seems very gloomy, I know, and I love this poem on the left-hand side by the Liverpool poet Roger McGough. It wasn't written about COVID, but it could equally apply. Every day I think about dying, disease, starvation, violence, terrorism, war, end of the world. It helps keep my mind off things, which sounds very gloomy. I find that strangely helpful. But even more helpful is the really positive, accelerated developments that have happened that really will stand us in good stead for the future. Just one of those is the vaccine development and speed and implementation and deployment of vaccines. And this is Sally Gilbert, who was really important, Dame Sally Gilbert, who was really important in the development of COVID vaccines, now saying, right, we've done this for COVID, let's do it for a number of other important diseases as well. And this is my very last slide. I've probably missed people off, and I really apologise if you don't see your name up there, but this is just to give an illustration of the people that I've worked with that have been involved in some of the work that I've described in this talk, and some of the funders that have also helped to support the work that we've been doing. So that's it from me, and I'm happy to take some questions. So I'll stop sharing, if that's okay.
Great, thanks Nigel, that's fantastic. I think we've got some mower in the background there somewhere. But,
yeah, sorry, that was the lawnmower that went past just at the end.
Oh well, life carries on. So that's really fantastic seminar. We've got one question from Arno Van Vlie in the UK. He's up there late at night. He says, genome sequencing gives us some great ways of understanding what's happened, like the black boxes in planes can do when there's been an accident. How can we make it more prospective, and what can we make predictions of what the likely next big step is going to be, accepting that we get it wrong regularly? Would it be the Rosetta Stone for pathogen epidemiology? Any thoughts on how we move towards that ideal situation?
Yeah, well, thanks, Arno. So Arno, for those of you who don't know, is on our International Science Advisory Panel for the Food Safety Centre, and is an internationally recognised expert in this field. So I probably should really turn the question back to him to answer that one. But he's also a very strong advocate for the application of genome sequencing and the potential of what it can do. And I think one of the things is we still struggle with our inability to predict when, where, and how pathogens are likely to emerge. And I think genome sequencing alone isn't going to solve that. We need to have really good prospective sampling of wildlife populations, of other potential host populations, combined with effective modelling approaches to really identify not only when and where pathogens have emerged from, but also when and where pathogens are likely to emerge in the future. And really our ability to do that really is a huge challenge, I think, for the global scientific effort in this area. I mean, I think one of the experiences that we've had, I think, have been encouraging with COVID in particular. We've seen a really increase in the turnaround time of genome sequencing from months to weeks to days, and sometimes now within 24 hours we can get a really good idea, for example, of where a mystery case is likely to have arisen from, or you know, to help actually inform decision making in real time. We have, or ESR have managed to sequence, I think, over 95% of all of the COVID cases in the current Delta outbreak, which I think is a great achievement. And it's really getting that high coverage, really high quality genome sequencing, rapid turnaround times, modeling, and other technologies as well, such as the use of non-culture based approaches, I think will really help us to really capitalize and make the most of the data that's generated through the development of models and other informatics tools, which can really help to form decision making in real time. I'm not sure I've answered your question very well there, Arno, but feel free to.
Well, Arno has commented, he can't speak here because we don't have a facility, but environmental surveillance and non-disease needs to be much more extensive to understand what's out there, and also how we can convert genome sequence data into risk analysis. Just on that time aspect, I wondered, I couldn't quite see from your slides around the bean sprout outbreak. How many weeks was that before it was actually discovered, or was it months? I couldn't see the timeline.
Well, the delay was in the time. Obviously there was an incubation period, so the period at which people were consuming the contaminated bean sprouts, so there was then a delay before the human cases started turning up. So you saw a delay of, and it was quite an extended incubation period. It was one of the unusual features of that outbreak, which actually is common to bean sprout outbreaks, is a relatively long incubation period. So that was several days before the peak, but then there was a lot of work. It did involve some genome sequencing at the time. There was crowd sourcing, or people were analyzing the genome sequences in places all around the world and contributing to our understanding of that. And there were diagnostic tests that were being developed. But really, by the time it was really found out what the problem was, the epidemic was already over. So. Yeah, several days.
I guess things have moved on a lot since those days anyway. It's a lot faster.
Yeah, I think the speed of turnaround is a key thing. And I think it's easier for viruses than it is for bacteria to do the very rapid 24-hour genome sequencing. But I think there's hope for the future for speeding up things.
Yeah. Another question I have, is there evidence in New Zealand for increased antimicrobial resistance, and what are the efforts that are being made to combat this?
Well, I showed an example, a Campylobacter example of emergence. I think New Zealand, we have got a number of strains that have emerged, particularly things like the extended spectrum beta-lactamase, E. coli. We did see an increase in resistance in Campylobacter. We do see some increase in resistances of particular pathogens. I think and ESR obviously doing routine surveillance on that. I mean, it is a concern. Our levels of antimicrobial resistance are not as bad as they are in many other countries around the world, but certainly it is a concern, and it's something that does need to be addressed, and it is the focus of the Prime Minister's Chief Science Advisors panel looking at AMR in New Zealand at the moment. There will be a report produced on that later in the year.
Thank you. I think that's all. I think we're sort of running out of time. People are starting to go, but thank you I'd just like to say thank you very much, Nigel. It was a fantastic presentation and seminar, which we will send out to people as after we've got the recording sorted. Appreciate you taking the time to give us such an excellent seminar, especially I know you've got a big workload at the moment. Just to let you all know that the next seminar, which will be our last for 2021, will be on usual time, Friday the 26th of November at 10 a.m. So we've got to sort out the subject and topic, but probably like to be an industry speaker. So really hope you can all come along to that too. So thank you very much again, Nigel, for a fantastic presentation. And thank you for joining us from all parts of the world, including Arno and I think someone from Australia. So thank you for joining us today, everyone.
Thank you.
Okay, thanks a lot.